This is episode 274 of the Stem Cell Podcast, Brain Organoids, with Dr. Ziyuan Guo. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Stem Cell Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, as our guest, we have Dr. Ziyuan Guo from the Cincinnati Children's Hospital. He's on the podcast to talk about his research on modeling the nervous system in a dish. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, looking to incorporate neural organoids into your research, get a free wall chart showing how neural organoids are generated and can be used to better understand brain development. To learn more, visit stemcell.com/brainorganoids-wall chart. And indeed, we're going to start things off with a couple of organoid papers in Nature, pretty high profile here. First one I'll talk about is a Nature article titled "A Human Autoimmune Organoid Model Reveals IL-7 Function in Celiac Disease." And this is coming from Calvin Kuo at Stanford University. First author here is Antonio Santos. Also Mark Davis and a couple other familiar faces on this particular paper. We're talking about celiac disease, which is what we know as, is actually quite prevalent these days. It's an autoimmune disease associated with gluten, where, you have this dietary gluten-derived peptides that can cause a lot of problems for folks who have this disease. So unfortunately, they can't have certain types of bread and that sort of thing, right? And so they're basically establishing an in vitro model of autoimmunity focused on this disease of celiac disease, right? It's, you have the inability to basically metabolize certain gluten molecules, right? So basically this dietary gluten-derived peptides bind to MHC complexes, initiating immune-mediated injury to the duodenum in the gut. So there haven't been a lot of these model systems. You can understand that this is something that's tough to model in vitro, although we have a couple of papers that are going to focus on this exact topic. And maybe that's why they're actually so high profile papers, is because it just has been so tough to incorporate this autoimmune aspect into these organoid models. You have to have the tissue of interest, of course, but then you have to incorporate the immune system into that organoid tissue. So here they pretty much did just that. They generated an air-liquid interface, or ALI, duodenal organoids from primary intact fragments of endoscopic biopsies. It's pretty cool because, people have endoscopies all the time, right? So you have an endoscopic biopsy creating these organoids. And the cool thing is, in the ideal organoid scenario, you're going to preserve that tissue architecture. So here they're preserving the epithelial alongside the native mesenchyme, and most importantly, the tissue resident immune cells as a whole unit without actually requiring additional reconstruction, or reconstitution. And they, in these organoids that comprise not only of the duodenum and the tissue there, but also the immune cells, the immune diversity was also retained. So you had T cells, B cells, B plasma and B and plasma cells, NK cells, myeloid cell, everything that you would normally expect in that immune niche, you were finding it in these organoids, and also extensive T cell and B cell receptor repertoires. So these gluten peptides were able to instigate epithelial destruction when they added them exogenously in HLA expressing organoids derived from the celiac disease patients. And the important thing is they ended up blocking this response as well, using by blocking the MHC complex, which kind of makes sense. So the gluten epitopes stimulated a celiac disease organoid immune response, sort of like what you would see in the patient, in the lymphoid and the myeloid subsets, alongside autoantibody production as well, which, again, something you might expect. So the important thing of these organoid models is they're ideally recapitulating the disease phenotypes, but something like autoimmune disease, you have to have the immune component in there. It's really cool to see that this is accurately and faithfully recapitulating that celiac disease phenotype. And then they went on a little bit to do some more functional studies in these celiac disease organoids, showing that actually IL-7 or interleukin 7 is a gluten inducible pathogenic modulator that's actually regulating T cell expression, is actually necessary and sufficient for epithelial destruction. So, again, coming back to a node in IL-7 that may be regulating this disease phenotype. And then when they added it, when they had endogenous IL-7, it was actually upregulated in patient biopsies from the active celiac disease compared with folks in remission. So it's like you got a biomarker here in IL-7 of perhaps active celiac disease compared with folks who are in remission from a gluten-free diet, which is the easiest way to kind of address celiac disease. But if you want to dive into the mechanism and if you want to figure out some potential therapies for it, you got to use some of these in vitro model systems like these really cool air liquid interface organoids. So it's, I think, a really great model. It's able to preserve the epithelium alongside those immune populations to faithfully recapitulate some of the disease phenotypes in a dish in this celiac disease organoid model. You have the gluten dependent pathology that you can, again, recapitulate here. And ultimately, it's useful for mechanistic investigation and mechanistic interrogation as to how this disease, which I think has become very prevalent, especially in the developed world, how this disease arises and some of its mechanistic underpinnings.
Yeah, this was a really cool study for me. Although, I mean, I didn't go as deep into it, so forgive me if I'm a bit off target here. But the thing that really was astonishing to me was the time scale that they were working on here. Approaching a year for some of these organoids in the air liquid interface, and I guess that's how the technical element here really it seems to have facilitated this insight and discovery, is getting a system where you can carry these things for a long, long while seems to be key to seeing the development of the phenotype and recapitulating all those elements there. But it was striking to me just because, when you take things out of the body, put it in like not static, but relatively static context of cell culture, it's remarkable that all these cells can coexist and maintain kind of their physiological function in the molecular biologies and cellular, coordination amongst all these types. For an extended period. But maybe I'm wrong there. Maybe they didn't have to go out to a year in order to start the experiment. I'm pretty sure they did not. But still, I mean, I think it's remarkable that all these cells can coexist in a in vitro environment, and clearly the air liquid interface was a key element to that.
Yeah, I think it was an important mesh of bioengineering plus organoid biology. But I think, especially when it comes to the intestinal tissues and gut tissues, perhaps these have been the most amenable to organoid creation, at least primary organoid creation. You talked about way back in the day with Hans Clever's creating some of the first LGR5 positive intestinal gut organoids. Perhaps it's the proliferative nature of these cells, how rapidly they can grow, and which makes them amenable to primary organoid creation. But I think the most critical part of this is the niche, right? You have the immune niche that's there. It's not lost just by growing these organoids for an extended period of time. And I think that's the key. That's the key why this is such a high profile paper. And actually, I'm kind of surprised that this hasn't been attempted before. Because as we know, the immune system is so important in regulating all sorts of diseases. And this is specifically focusing on autoimmune disease. So if you want to model celiac disease, you have to have that component, right?
Absolutely. And I mean, this is, I think, reflective of what's going on broadly speaking in, Assembloids are a great example of it. And this isn't exactly that because, as you said, I mean, it seems to be, I wouldn't call it low-hanging fruit. I think it's still technically a great challenge, but you're just taking it out with all the components in there and then kind of throwing it into culture and letting them all coexist, as opposed to these approaches, I'd say more broadly speaking, that start from individual cell types that then you integrate together. But as you're saying, I mean, it's a key finding, and I think a key technical advance to be able to approach greater complexity with these organoids. Assembloids being a great example, as I said, but also I think getting diverse cell types and systems, namely hematopoietic and immune cells, so you can understand some of these autoimmune things. Because yeah, organoids typically, ever since early days, those LGR5 cells that you mentioned, that we're talking about epithelial cell types. We're talking about uh, by and large, epithelial only structures, what these organoids, in order to take on the architecture, that's epithelial. Whereas, of course, the correlate that the organoids are trying to approximate are comprised of many different cell types, most notably, perhaps, immune cells, specialized immune cells. And this is really exemplified by the intestinal mucosal immune system, that founder organoid, here now being expanded into something that can be more clinically useful. And that's the segue into my story here. As you said, a Nature story 2 weeks after the one you just mentioned. So kind of a twofer here. And yeah, talking about that intestinal mucosal system in this case, and generating more complex organoids that incorporate that immune component, because disruption of that intestinal immune function leads to persistent infections, autoimmune conditions that we're talking about, like celiac, even malignant disease. And while intestinal organoids from day one, right, the first organoid, are able to model a lot of key aspects of the epithelial cell types present, they fall short in recapitulating the intestinal pathophysiology that relates to immune function. So to address this, I mean, this is a parallel, I would say, approach to what you just described from QuoLab. This is a group from Roche, the Institute of Human Biology. It's three leads here: Lorian Cabon, Jay Gray Camp, and Nikolcha Jorevsky. Excuse any mispronunciation. My apologies there. But this group at Roche, they, in a similar vein as you, they created this intestinal immunoorganoid model, they call it IIO model, that contained the tissue resident and here key autologous immune compartment, just like you're talking about here, getting it from primary patient samples, like it's all there to begin with. But here they use a different approach, which wasn't just that they I don't want to oversimplify. It's all very complex. They didn't just throw it in the culture in the Quo study, but here they had a different approach from transposing it into culture. Maybe that's just a euphemism for throwing it into culture. But nevertheless, they transpose these things into culture. But first, because these tissue resident memory cells, these T cells, that are the target population in terms of their immune effector function here, they don't really survive enzymatic dissociation. So what they did, and I thought this is so cool and so underscores how, the most complex science sometimes is, I don't know, kind of low tech. Here they use this migratory, scaffold-driven migratory approach where they let these resident memory cells like crawl out of the biopsy, and then they froze them separately, isolated the epithelial cells that were resident in the biopsy, and then generated organoids out of those over the course of 2 weeks. Then they thawed those tissue resident memory cells and reincorporated them into these organoids. And this story was kind of a proof of principle, but I think a useful one here. They validated all the cell states in these intestinal immunoorganoid model, comparing them to these reference atlases, single cell seq. And then they showed that these tissue resident memory cells, they integrated within the epithelium of the organoids and continuously surveyed the barrier. They also used single cell transcriptomics to show that inflammation was triggered by these cancer targeting biologics that are implicated in some of the side effects of these new cancer treatments. And they showed kind of sequentially, looking at the cell level, showing that this inflammation, it first started cause the activation of these CD8 positive T cells that acquired these cytotoxic features. And they showed that even before that, there was a kind of initiating cell population, these CD4 positive cells that produced a lot of cytokines that activated that CD8 positive set and then ultimately became cytotoxic in their own. And then finally, in terms of like therapeutic intervention here, they identified this Rho pathway implicated in this sequence of events and therefore identified as a target for maybe potentially mitigating the intestinal inflammatory side effects that follow from immunotherapy. So, for me, I love a study like this. Number one, shows pharma's going deep on the basic science here to establish these models, and showing that it really can be applied in a way that's useful and has promise, not just for modeling this immune organoid interface, but actually identifying targets that can have therapeutic implications in the context here of cancer treatment. So a really nice study, and just the little things again, just to reemphasize how how you get over the technical hurdle here of low survival of T cells from enzymatic dissociation by generating these hacks. Every great science story, I think, has a bit of a hack in it. And this, as a complement to the quotes that you just described, shows how we're hacking organoids in order to model the immune interface. So really exciting series, a couple of stories that we're talking about here on the show.
Yeah, two sides of the same coin here. I mean, we're talking about immunology. Hopefully we're doing justice to this, and shout out to the immunology podcast. Maybe they'll be covering these stories on their roundup this week as well. Yeah, these are two kind of approaches to model the same thing. Broadly speaking, you're trying to model the immune system and the impact of the immune system in this organoid context. The first one was autoimmunity and looking at celiac disease. This is focused a little bit more on potentially drug toxicity, which is why I actually really like this story. The other part of this that's very different is the timescale of the model system, and maybe you alluded to it on the first paper. There was a multi-month long organoid prep, 1 year long organoid prep to actually make those celiac disease organoids. Here you're just talking about a couple weeks to make these things and ultimately just freeze them down and then scale them up for downstream applications, which is great. But again, like both stories have the same focal concept of modeling the immune system in the organoid context. As somebody who studies drug toxicity, focused on cardiotoxicity, I thought this was really an interesting study to look at and consider, because immunotherapy unfortunately has a lot of side effects, and we need model systems to better understand how immunotherapy impacts, say the gut, or in my case, the heart. We know there's immunotherapy associated myocarditis, which is a huge problem with a lot of the recent immunotherapies that have been developed out there. And unfortunately, the mouse models haven't been great model systems to study these off-target effects of immunotherapy. So that's where these newfangled organoid systems come into play, right?
Yeah, and all different types, right? The long temporal scale you described, and the shorter one here with different costs and benefit. In the short scale, I like the idea again that they got the hack, they got them out, and then they froze them, so they are kind of I would guess representative of that time of point of origin, right? And frozen in time, as opposed to some of the kind of phenotypic drift or non-physiologic function that you might expect any cell, but hematopoietic and immune cells in particular might take on over the course of these long time scales. So yeah, I mean, I think as we're both saying here, kicking open the door to this immune modulatory influence in the context of drug toxicity and autoimmune. I mean, it's interesting the timing of these things. It wasn't the same issue, but as we've seen before, it seems like the time is right, and the pace of innovation converges on very similar stories. And here, I think there's going to be a lot more stories to follow from this type of approach.
Time is money, but I think one set of folks who are definitely in favor of the short time scale and the 2-week organoid as opposed to the 1-year organoid are your PhD students and your postdocs who are actually doing the experiments. I mean, if you tell them that, hey, we can do a 2-week long differentiation, make some organoids, scale them up and freeze them and then use them for later, maybe there's an advantage to that. Maybe that's a little bit more attractive than a 1-year long organoid culture. I'm just saying. Moving on to something a little different, but still in the organoid-esque, organoid tangential space. This is actually a Nature Materials paper, and we don't talk about Nature Materials too often. I mean, that's like it's more of an engineering-oriented journal, but a lot of cool stuff in there. And this is a Nature Materials study intersecting with early developmental modeling, something that we talk a lot about here on the show. The title is Substrates Mimicking the Blastocyst Geometry Revert Pluripotent Stem Cells to Naivety. A very, very hot topic, this naive state that everybody's striving to get with perhaps a higher state of potency, more amenable to differentiation, more amenable to downstream applications, become a very hot topic recently. And if you haven't been living under a rock, you know that we've been covering this extensively here on the show. So naive pluripotent stem cells, right, or NPSCs, they have this really high developmental potential, but their in vivo existence in the blastocyst is pretty transient. You got to catch them at the right time. And here they're doing a really cool engineering-oriented approach to maintain and extend the lifespan of those naive pluripotent stem cells in both mouse and human. So they're looking at a blastocyst motif substrate for the in vitro reversion of mouse and human pluripotent stem cells to a naive state. So you find the right substrate and you can make these pluripotent stem cells stay naive for longer. And the substrate is, it's basically taking advantage of geometry. It has these randomly varied microstructures, which they call motifs, that are mimicking the geometry of the blastocyst. And they have to do some really cool modeling to really figure this out. Take a look at this paper. A lot of computational analysis early on to drive the downstream. So the motifs representing the mouse blastocyst scaled curvature were ranging between 15 and 62 millimeters. Again, they had to look at a lot of individual blastocysts to feed this data set of figuring out what's the right motif, what's the right geometry to actually make this happen. So stuff that was between 15 and 62 millimeters was the most efficient in promoting reversion to naivety as determined by time-resolved corrective analysis. And in these substrates, the apical constriction actually enhanced E-cadherin and RAC1 signaling. So this is how they brought it back to the molecular mechanisms. They dove deep into not just saying, so they went beyond just saying, okay, we got a substrate and it's able to extend naive potency. They wanted to figure out how that's happening, right? So the substrate was actually enhancing E-cadherin RAC1 signaling. And something I thought was really cool, enhanced and activated the mechanosensitive receptor nucleotransducer YAP, which we have talked about on the show. Of course, YAP has this really cool phenotype in enhancing proliferation in the cardiac context. YAP mutant mice have massively hypertrophied hearts. It's one of the most dramatic phenotypes I've ever seen. It's very critical for regulating cell proliferation and cell fate. So YAP also had this node role in promoting the histone modification of pluripotency genes and ultimately resulting in enhanced levels of certain pluripotency factors that we all know and love, like NANOG. So just by growing these cells, these pluripotent stem cells on this substrate, you're ultimately facilitating the enhancement of the NANOG signaling pathway, which, get this persisted even after the cells were removed from the substrate. So it's almost like the substrate just sets them on the right path, and you can take off the substrate and the cells will still retain their enhanced level of potency in their naive state. So these pluripotent stem cells cultured in the blastocyst motif substrates developed a higher developmental potential in actually generating better and more substantial embryo bodies, as well as teratomas, which is something that you can use to validate the potential of these naive state cells. So it's a really cool study intersecting engineering with developmental biology, shedding light on how substrate design can perhaps enhance the maintenance of these naive states. And the other cool part of it, which they allude to at the end, was how you could use this in potentially large scale. So if you want to really scale up the production of your naive pluripotent stem cells, maybe this substrate is the key.
Yeah, shape and fate have been, there's a long history, right? Like Mina Basel back in the days that the mammary cells would make casein only when they were 3D. And then I can remember it's almost, I had to look this up, almost 20 years ago that Engler et al. And Cell uh, showed that the matrix elasticity dictated the naive mesenchymal cell fate, right? Which was a real breakthrough. And since then, as you talked about there, the YAP and incorporation of architecture as a key mediator of cell fate and appreciation of that has really grown. And we're in this era now, right? Remember ISSCR between, the Lutoff microchannels to make the gut mini guts. We had some neural tube. We talked about on the show a little while ago about the segmentoids and somatoids. So, the idea of either having a templated architecture or just the constraints of shape of cells relating to fate. There's been, I would say, a deep recent history of that, and this is another great example of that. And it makes you appreciate again, how do you model this? When I saw the title, I'm picturing them, they're seeding cells into these tiny little balls. But no, it's the way that they create a substrate with this, variable convex and concavity and all these things. The way they get at it again here in this methods and the way they model it, I found so fascinating. And this has really important insight here. In this case, showing that you could revert a cell, right? It wasn't instructing a cell in differentiation, taking a pluripotent cell forward or some kind of progenitor forward. It was the idea that if you constrict or incorporate the cells into a predefined architecture, that they can revert. So I think this is, in terms of conceptual insight, raises a lot of questions about how we optimize everything that we do in cell culture, depending on the purpose. But I can envision a future where the substrate and the microarchitecture there, it's already there. We're already talking about it. With these micro etched planes to pre-pattern, et cetera. But I could see how this is becoming not just like, a feature of cells, but almost an important prerequisite to cells actually taking on the fate that they are directed toward. Is that you may want to incorporate the substrate modifications in this way across the board, which is, I think, sounds really laborious now, but we may just have plates for every kind of diff. And imagine a world where, the substrate was as important as the cytokine, or maybe made it a lot easier to do these diffs.
Yeah, it's something we forget a lot about when we're talking about in vitro culture is, our systems are entirely artificial when it comes to the substrate. We're just most of the time growing cells on plastic dishes, which is not analogous in terms of the stiffness and all that the cells would experience in the body. And of course, there's been a lot of work on this and trying to modulate the stiffness of the substrate to influence differentiation, as you alluded to. But part of the reason I really like this paper is they looked at the opposite. They looked at the reversion, the reprogramming, the demonstration of the naive state and how that's influenced by the substrate. And they found something really cool. This big field of mechanotransduction has been around for a while now, but I think it's really starting to get a lot of love in stem cell biology. This is a case example of it. course, mechanotransduction has been a very hot topic. And for example, Artem Patapoutian received the Nobel Prize not too long ago in the field of medicine in 2021 for his work on the piezo channels, which are also very important for mechanotransduction. We just talked about what happens when you take out an element of mechanotransduction and gravity entirely from the equation. We had our recent show on stem cells in space. So mechanotransduction is influencing everything. Don't forget about the stiffness of your substrates, and don't forget about the influence of gravity either.
Yeah, I mean, because that's a great fallacy from day one. I mean, there's these pictures of the homunculus. If you're ever seeing the what people thought about human development, they thought there was a tiny little man, a person, homunculus that lived in the sperm head. And I guess that's the great fallacy that has kind of continued on into even the modern days, this notion of intrinsic capacity, when the reality is that we're all a product of our environment at the macro and the micro scale, of course. And then the next term I'm going to talk about is like in terms of like cellular capacity. Here we're talking about neuron excitability, action potential, right? I mean, that's the defining characteristic of neurons is action potential, excitability. They transmit a signal, right? And in fact, that's the feature of a lot of the pathology, neuropathology, dysregulation of excitability, whether it's chronic pain, epilepsy, even neuropsychiatric disease, amongst other things. It's about excitability and defects in that action potential. And during development, we talk about this, of course, in terms of maturation of all cell types, but specifically in neurons. Neuronal maturation is marked by the acquisition of proper electrical function, right? But the way that these neurons acquire that excitable potential during development, it's not really understood. I would say, if you asked me before I read this story, I would just say, hey, they're neurons. That's what they do. It's like, tell me how a heart acquires its contractile, or cardiomyocyte acquires its contractile properties. You just assume it's intrinsic to the identity of the cell. But somatosensory neurons that innervate peripheral tissues to transmit the sensory inputs. Initially, those neurons, after they're specified in development, initially, they're not excitable, right? They only become excitable upon expression of these voltage-gated sodium channels. And then from late embryogenesis until after birth, those neurons further mature to become fully responsive to sensory stimuli, right? But that transition from inexcitable to excitable and the expression of voltage-gated sodium channels, I, for one, never thought that was like an instructed thing. I thought there was a cell intrinsic capacity. They become neurons, and so they express these voltage-gated channels. But there's an idea out there that perhaps that isn't or it isn't at all a cell intrinsic process, or in part maybe non-cell autonomous, and that is driven by the observation that neuron excitability in development coincides with the first appearance of Schwann cell, or these glial precursors, right? And because glia are active regulators of a lot of important aspects of nervous system function, and in development, it led to this idea that perhaps these Schwann cells may contribute to the acquisition of excitability amongst neurons. And this is the question that was asked by J. Bradley Zucchero's group at Stanford University, is whether these glial cells dictate that excitable acquisition of excitability, and if so, how do they do it? So this was a story in Cell just from a couple weeks ago, and what they found was that, yeah, indeed, it is these Schwann cells that dictate. They showed in this, they made these highly purified cultures of just these kind of nascent neurons from the dorsal root ganglia. And they showed that when they isolated them, so they didn't have the Schwann cells with them, they were hypoexcitable and didn't really take on these actual action potentials. But if you added back just conditioned media from Schwann cells, that was sufficient to get both expression of these voltage-gated sodium channels and to generate action potentials. They went on to show that prostaglandin E2, so a factor that's been mostly implicated in the context of neural inflammation in adult and inflammation in other contexts, they show that that was secreted by Schwann cells and was essential to that acquisition of excitability by genetically ablating the signaling pathways, specifically in Schwann cells. They showed this neurodevelopmental defect that persisted into adulthood even. With these, I always wonder when I read this, like, how do they know about behavioral elements here? In this case, they do this heat withdrawal. So they put the foot of these poor mice on a hot plate, and the ones that had their PGE, prostaglandin E2, knocked out in Schwann cells, leading to a defect in that somatosensory neuron excitability, they had a latency. It took them 4 seconds longer for them to withdraw their paw, and other metrics like that, including deep single cell RNA-seq, profiling these cells and showing at the electrical level that they had these defects in excitability. So, I mean, for me, I think this is a exciting conceptual advance more than anything. I mean, that the implications for disease are obviously there, and I think profound in terms of just the relationship of this inflammatory molecule to the acquisition of full neural excitability and action potential function. Essentially, I think that's key. But more than that, just this idea that there's a non-cell autonomous mechanism that drives this seemingly intrinsic, a cardinal feature of a cell type is non-cell autonomous. I think that raises important questions about the function of every cell that we think of and the relationship of environment to the acquisition of cell fate, as we just talked about in your blastocyst story, or the reversion story with the blastocyst substrate, but also, intrinsic properties of the cell, not just the fate of the cell writ large, but the intrinsic properties of the cell, oftentimes that you think are just intrinsic, may be dictated by a partnership.
Yeah, not to be a boring cardiac biologist here again on the show, but as soon as you started talking about this, the parallel in my mind was obvious. Another highly excitable cell type, electrophysiologically active cell type that has this intrinsic action potential, is a cardiomyocyte in the heart. And you think about the first heartbeat, the first time those cardiomyocytes actually contract, the thought is that it's an intrinsic effect. And you even see it in cell culture. When I make iPS cardiomyocytes, they start beating on their own, or you'd like to think so, right? But then you start wondering, what's the influence of the other cell types, those support cell types, like the glial cells that you're referring to here, Schwann cells. The heart has the analogs too, the support fibroblasts. Are those playing an influential role in initiating that initial contraction? And is cardiomyocyte contraction not autonomous? I don't know. It's going to keep me up at night, Dylan.
Well, I hope you don't lose sleep over it, but I mean, I'm thinking along a similar vein here, right? I mean, it's not just, there's this idea of immaturity. We always talk about as immature cell type. That's the problem with any pluripotent cells derived. Well, I mean, this may speak to that. Also, just the notion of getting any highly purified cell type. We're fixated on 100% diff, right? But maybe we need some accessory cells there to confer a truly mature function, and that interrelationship, I think, has been it's a bit murky right now. But this is one story, I hope, among many to come, that will fill in the blanks there. So, a really exciting series of papers on the Roundup today. A lot to talk about. Perhaps we can parlay some of these ideas into the conversation with Dr. Guo. Before we get there, though, I have a quick message from STEMCELL Technologies. If you use organoids, have a look at the StemDiff Cerebral Organoid Differentiation Kit by STEMCELL Technologies. This 3D culture kit reliably mimics early brain development, so you can focus on your next questions instead of troubleshooting. The possibilities are endless, and the future begins now. Learn more at www.stemcell.com/cerebral organoids. All right, everybody, for this episode from Cincinnati Children's Hospital Medical Center, we have a special guest, Dr. Ziyuan Guo, who is assistant professor in the Department of Developmental Biology and also at the Center for Stem Cell and Organoid Medicine there at Cincinnati Children's. The Guo Lab is working to develop a next-generation human nervous system in a dish using cutting-edge stem cell technologies and engineering tools. In his lab, they apply innovative stem cell technologies to study the etiology of neurodevelopmental and neurodegenerative disorders in the context of human genetics. Ziyuan, thank you so much for joining us on the show today.
Thank you for having me here.
Yeah, it's great to have you here. And like Dylan said, your lab focuses on all things neurodevelopmental modeling, neurological modeling with iPSCs, and you've been in this field for a while. You're using all the coolest tools in the stem cell field, like cerebral organoids, CRISPR, to really figure out the fundamental mechanisms of neurodevelopmental disorders. So let's start things off with a broad overview, in your words, of what your lab is working on. I mean, we gave the summary, but we'd love to get your perspective on what you do and maybe a little bit about how you actually got into this field in the first place.
Well, of course. So actually, trained by the neurodevelopmental biologist at the Penn State when I was come to the U.S. To pursue my PhD degree. So, well, actually, I'm the first one, and which is the first lab, of course, at the Penn State to develop this transdifferentiation technology, or you can call them in vivo reprogramming. So basically, we can use the pro-neurogenic factors such as NeuroD1 to convert the reactive glial cells into functional neuron in adult brain for the brain repair. And after that, actually, I joined the Hongjun Song and Guo Liming's lab as the UPenn now. And actually, when they was Hopkins, I joined their lab. And actually, I trained as the stem cell and organoid medicine scientist at that time. And we are using a variety of the patient-derived iPS to study the schizophrenia. And later on, I started my lab at the Cincinnati Children's Hospital in 2019, just right before the COVID. And I know that's tough time, but we are successful to set up the lab. Now my lab actually are focused on three main research avenues. So first, of course, we are using a lot of patient-derived induced pluripotent stem cell to study the disease mechanism in neurodevelopment disorders such as autism or degenerative disorders such as Alzheimer's disease. And recently, we developed this very cool, the human blood-brain barrier assembloid by, combining the human brain organoid and a blood vessel organoid to mimic the neurovascular co-development and develop the first, we believe, the most authentic, the human BBB structure in the organoid culture system. And we also continue like this in vivo reprogramming approach that we hope this highly translational approach can be sometime in the future can be used for the patients.
Yeah, man, you're casting a very wide net, shooting at a lot of targets and hitting them for the most part. But it's a wide net. I mean, neurodevelopmental and neurodegenerative conditions cover a pretty wide spectrum of unmet medical need. And of course, the approach for addressing each of those conditions, highly disease specific, even patient specific in a lot of cases, right? So I'll ask you, as someone who's really thought about this deeply and is shooting for the stars, with all these, not all these, but we got multiple cell-based therapies that are in clinical trial in the neural space, right? It's a very exciting time for our field generally. I think, stem cells, cell-based therapy generally, but specifically in neuro. What do you think are the greatest challenges facing the realization of that long-held dream of cell-based therapies in the brain?
Well, yeah, I think that's an excellent question. Well, it's $1 million question as well. The cell therapy that's actually a lot of scientists in the past year to develop a specific for example, the Parkinson disease. We have a successful generate a very pure, highly pure the dopaminergic neuron and the transplant in the patient. Then we hope to get some really beneficial impact for the patients. Well, I think the whole scientific field already make a lot of successful, but how it can be a benefit to the patient, especially in the long term, the therapy treatment windows that we still don't know yet. And specifically for this neurodegenerative or neurological disorders, this our brains are such a unique, organs, right? So it's not only that you replace the neuron, then everything done. Actually, it's really how this the cells that you transplant can really talk to their neighbors and correctly in the right way, in the endogenous the cells way. For example, for Alzheimer's disease, if you just replace the lost neuron, can you really recover that memory? That we don't know yet. So, but anyways, over a offer a new avenues for the whole world that we can so far use different approach, not only cell therapy, but also the gene therapy. As I mentioned at the beginning, for the in vivo reprogramming, we can convert the endogenous glial cells or reactive glial cells into functional neuron to at least rejuvenate this neuron generation in adult aging brain that really maybe someday when we figure out how to make the right cells, make the right connection, then we can eventually to help this patient enormously.
Yeah, so actually let's talk about that approach, that direct reprogramming approach. That was really the kind of the foundation of some of your lab's work. And you had that stem cell paper that we talked about that happened about a decade ago, turning functional glial cells, reactive glial cells into functional neurons, like you alluded to in both this brain injury model and the Alzheimer's model. I mean, this approach of direct reprogramming in vivo, we've talked about it before on the show. And for example, I'm a cardiac biologist, so I know that Deepak Srivastava's lab is also attempting something similar in the heart, trying to turn fibroblasts into functional cardiomyocytes. Easier said than done sometimes. It's the efficiency of the process has been kind of mixed, I know, but it definitely has a lot of potential, like what you're alluding to. So why don't we talk a little bit more about that direct reprogramming approach, especially in vivo, and maybe some of the challenges that are still associated with that approach?
Yeah, of course. Yeah, I really like this. I think it's a very promising, therapeutic and translational potential, this approach. We call them glial to neuron conversion, or you can call them as a in vivo reprogramming, transdifferentiation. That's all okay. So the principle is how we can for the induced pluripotent stem cell, basically you use the Yamanaka factor or other approach that to turn the somatic cells back to the pluripotent stage. However, for this transdifferentiation is slightly different. So basically, we and other lab to develop to discover the different pioneer factor that can efficiently and powerfully to convert one somatic cells into another somatic cells. In our case, it's from the astrocyte or microglia, basically into the functional neuron. So this principle, the first as actually discovered by the Marius Wernig. I think you guys already covered a lot of his story as well. So, but we are the first one actually can sufficiently and to convert the endogenous astrocyte and the reactive astrocyte into functional neuron. And of course, you can imagine, in a lot of neurological disorder, including the brain injury, spinal cord injury, stroke, or neurodegenerative disorder, the first challenge is how to regenerate this lost neuron, right? So that one, as everybody knows, before we develop this approach, once you lost the neuron, it's very hard to generate them, especially in adult brain. So, well, for our approach, we have actually one stone for two birds, benefits. Basically, we can convert those glial scar, the reactive glial cells, which initially prevented the damage in the brain, but later on, they become to some barrier for the neuron regeneration, axon regeneration. So our approach is we can convert those the reactive glial cells in the glial scar into functional neuron. So when we generate the new neuron, and meanwhile, we actually can improve this glial health as well. So we convert this reactive glial cells, we actually repopulate the remaining astrocyte to become more healthy microenvironment for the the neural regeneration. Of course, it sounds like a very beneficial approach. However, in terms of how it can help the patient, it's still a lot of things we need to overcome, right? So the people, the whole scientific field ask a lot of questions about, is this the new neuron generated is the same with this aging neuron? Are they still have issue about neurodegenerative phenotypes, or are they going to die as well as other like neighbor cells? Where actually our approach is also another benefit. I believe this pioneer factor like NeuroD1 can not only regenerate the neuron, but also it somehow rejuvenate the cells, endogenously. And another important question, as I mentioned, is this new neuron born can make the right connection, right? So for example, for Alzheimer's disease, this new neuron born, can they recover this memory that are lost already in the patient? That's very challenging that we don't know so far yet. So for other issue, of course, our technology can overcome some like a cell therapy challenge, for example, the immunoresistance, because the cells has been born just in the brain, the same region. But in terms of the long-term survival of the newborn neuron, that's another very tough question, whether we can maintain, we can efficiently generate those new neurons, and whether those neurons can survive very well in that like a disease environment for long term, right? So yeah, so we are working on towards that direction, but I think that's really a new hope for the patients. Yeah.
Yeah, I have always been so excited about this direct reprogramming, direct conversion approach, because yeah, as you mentioned there, all the benefits that address some of the outstanding challenges to cell therapy, right? The immune barrier, the delivery of cells, they're already there, right? And as you mentioned, sometimes in a highly elegant solution like your own, you can take the bad cells, the cells that would more normally have like a more maladaptive function, and convert them to something that's more constructive and regenerative. And similarly in the heart, same idea, right? Take that kind of scar and turn it into muscle. But as Arun said, it was 10 years ago, and there was a wave. I mean, I remember direct reprogramming, like everything in science, organoids or, IPS, different recipes for reprogramming. It's there's always trends, and a field gets really hot. And then there's like a latency, of course, before the hot trend moves on to something else. But people like yourself are still grinding in the lab to actually bring that exciting new finding to something that's really practical and clinically translatable. And I guess that's what accounts for the years that it takes before we actually see translation. But in that same span, right, 10 years, I would say one of the major things that have changed in terms of our technical capacity is the resolution, right? The single cell. So I know you just went deep on all the challenges, and maybe this is just a basic, straightforward question, but it really has been a huge point of curiosity to me, because now we have all this capacity to revisit some of the questions that maybe we couldn't answer or didn't have the resources to answer practically a decade ago. What do you think in terms of like enforced expression and taking a cell that was on one developmental or one track, at a mature level, and then converting it? Like, do those cells, how closely do they approximate the cell that you're targeting? Like, if you were to do single cell seq on these cells in situ that were converted, do you think that there's a high degree of fidelity in terms of how well they're able to arrive at like a bona fide neural identity? Or maybe you've even done those experiments. Can you just weigh in on that for me?
Yeah, I think you really hit the point, the excellent point. I believe my lab and maybe many other labs still doing the similar study or experiment. So leverage all of this advanced technology, as you mentioned, the single cell or single omics approach. We are looking into the detail deeply about this newly reprogrammed cells in the culture and in vivo. So of course, the one of the important question that is this newly born neuron identical to their neighbors, or if it's not, what's the difference? Is it different? It's good or bad, right? So we'll look at that. So I couldn't give you like 100% the question yet, but at least so far, our examination by use the single cell omic or other approach, even for the traditional immunostaining like a marker expression, we do feel the reprogrammed cells, because of this microenvironment for other cell interaction, their factor relief by their neighbors, they are able to acquire some of these region-specific identity, the neural identities. However, they're still not exactly the same with their endogenous cells. It may be because of the, this pioneer factor is not like a fully phenocopy, their developmental process and other additional factors may be required. That's one possible. And another possible is just simply because this is transdifferentiated cells, right? So they are already over the developmental trajectory. They are just directly become to neuron. They lose something, but they also gain something that we don't know from the epigenetic memory, etc. But the good thing is that the good side for this difference is it looks like those newborn neuron or reprogrammed neuron are more like looks younger, looks healthy than their neighbor disease neuron. Specifically for example, Huntington disease that we also published a Nature Communication paper talking about that, or Alzheimer's disease, we look at those newborn neuron, they looks more healthy. So which is good, right? So maybe because they are just newborn, so they don't accumulate a lot of the insult because of the disease environment. Or maybe this is a good side that can improve the brain health eventually for these patients. So, but again, this is, with this more and more new powerful technology, single cell transcriptome, and also spatial transcriptome that we recently use a lot, will be eventually help us to understand step by step, right? And spatially, temporally resolution to look at this newly converted neuron, how it has been converted and how this can be resemble their local, regional identities. And this is really in the future, maybe help us to understand, can they really replace those lost neurons in our system?
Yeah, a lot of interesting questions, and a lot of these questions are not exclusive to the direct reprogramming field. This concept of immaturity is, of course, something that we have to tackle in the iPS-derived neuron and iPS-derived cardiomyocyte field too, as well. I know that sometimes there's two sets of scientists, it seems like, in the stem cell field, one who are pro direct reprogramming and one who are pro IPS differentiation. And it seems like sometimes, there's some tension between the two groups, but I'm a fan of both approaches. I think they're really important parallel approaches to be taking. So shifting gears a little bit, you mentioned the second half of kind of what your lab is doing more recently, which is, looking at the blood-brain barrier and perhaps modeling the blood-brain barrier using some cerebral organoid approaches. You had the cell stem cell paper that you alluded to that we actually briefly covered here on the show not too long ago, modeling the formation of the blood-brain barrier in cerebral cavernous malformations and human pluripotent stem cell-derived organoids. I mean, this is a really cool application of this assembloid technology that we've covered here on the show. Like we've had Sergio Pasco here on the show many, many times. But here you're creating blood vessel organoids separate from the cerebral organoids and then just kind of mashing them together, combining them to create these blood-brain barrier assembloids. I mean, you alluded to at the beginning, there are some other folks who have attempted modeling the blood-brain barrier in other approaches. And I'm at Cedars-Sinai. We actually had a blood-brain barrier on a chip approach that we published a while back. But tell us about this assembloid approach and why it might be an improved way to study the function and the development of the blood-brain barrier.
Sure, and my pleasure. So so of course, the blood-brain barrier is such an important and unique, structure in our brain. So in the past years, a lot of brain organoid technology has been developed, including like Sergio Pascal, they are developed the Assembloid technology. However, of this organoid, existing organoid system is still missing the K brain vasculature and the blood-brain barrier structure, and especially to mimic how this neurovascular co-development process. And you mentioned your lab and many others has also developed like a BBB chip and different co-culture models. I think that's all very useful. But in terms of how to faithfully resemble this human BBB formation to decipher this underlying the molecular and the signaling pathway to guide this human BBB formation, and use this authentic the BBB model to study disease, which is still remaining elusive in the whole field. So our idea, just simply, we want to have a model that can really faithfully recapitulate during the early brain development together with the brain vascularization process. So then the idea, come from, if we look at the early human brain development in the late of the first trimester, that we have this perivascular plaques growing together with neurovascular cells and start to vascularize the neural tissue. Then we decide why not we just grow in separately the brain organoid versus the blood vessel organoid, assemble them together. Amazingly, we use this bioengineering approach to make the assembloid in a matrix jaw blade. We observe like a day by day. We say this labeling the vascular cells are just vascularize the whole brain organoid step by step, and eventually they form a assembloid. So during this process, we examine their gene expression, marker expression. We saw those naive, this blood vessel organoid gradually acquire this brain-specific signature, such as some glucose transporter or the PGP, the drug transporter expression, and also the tight junction, right? So this tight junction is a very important structure, they are different with other organ endothelial cells that to form the blood-brain barrier, low permeability and high resistance for their functionality. And then, of course, we are decipher into what kind of a molecular pathway during this process, and driven by the neural tissue to guide these endothelial cells to acquire the brain-specific signature. And eventually we find, okay, it's because of the Wnt signaling pathway, and we tested, added the Wnt signaling pathway. We actually, without the neural tissue, we still can induce those endothelial cells to acquire some brain-specific endothelial signatures. So this is a really model that mimic the neurovascular co-development, how the human BBB formation. Then you can imagine, use this model, you can either to better understand how the human BBB development, which maybe share some similarity with the other rodent model, but maybe some difference, right? So because we know the human and the rodent BBB, there are slight differences. So I think that's one of the critical reasons why so many CNS drug has been developed, rely on the animal model, fail in the clinical trial. That's maybe one of the reasons. So that's why we need to develop the human BBB model. And meanwhile, we can use this patient-derived BBB assembloid to understand betterly what happened during this BBB formation. Maybe even before the disease onset, the BBB already have an issue, right? So because the breakdown of the BBB has been observed in many neurological disorder, right? So that's basically a very common, the pathological hallmarks for neurodegenerative and the neurological disorder. Yeah, so that's why we developed this new approach to mimic the neurovascular co-development, BBB formation, and with this model, we can better understand how the human BBB formation.
Yes, we were very excited about this story. I mean, it's a great story. We reported on the show, but I was particularly enthusiastic as someone who has deep roots in Arun as well. I mean, he's been looking at the BBB and working at Cedars, where it's a specialty there. But I have also been very invested in the vasculature throughout my career. And yeah, we're living in this era of assembloids and all kinds of systems that are really increasing the complexity of our systems and models. In this case, what I really loved is, yeah, you recapitulating that kind of pathological hallmark from that anatomical standpoint, the intersection of the neuron and the vessel in this disease state. But of course, whenever I think of vessels, I also think of the blood and hematopoietic cells, and a huge component of some of the most, devastating neurodegenerative conditions is the neuroinflammatory component, right? So I immediately go to the idea of trying to incorporate immune cells like microglia into these model systems. I don't know, assembloid or not, to try and approximate some of the kind of neuroinflammatory conditions and maybe seek out some mechanisms to mitigate that. So is that like even feasible? Is that something, do you think you need to go in vivo in a kind of xenograft type model, or do you think that you can ultimately incorporate without maybe even vascular flow this inflammatory component into these systems? Is that a possibility? Did you see any microglia in those CCM assembloids?
Well, yeah, of course, this is an excellent, you basically hit the point that we are working on in many directions, basically. We are trying to, we don't observe any development microglia because it's come from different lineage, right? So based on we assemble just cerebral organoid and blood vessel organoid. So we are trying, a couple of ways. I totally agree. The immune system is such an important, especially if you have a BBB breakdown in the disease condition, how this can be contribute to the disease progress, especially in the like a stroke, brain injury, spinal cord injury, or neurodegenerative disorder. So well, there's a multiple way we're trying. So basically, we're trying first to incorporate immune cells and microglial cells in more complicated co-culture system with this BBB together with microglia. It's turned out it's not an easy task. So I can tell you because they are all different type of cells. They have different favor like supplements, growth factor. We are still trying to figure out what's the best the recipe for everybody can tolerate basically. So even well, I didn't mention this like a technical detail, but when we developed the first BBB assembloid, we have a similar issue, right? The endothelial, how you can make sure the endothelial cells and the neural cells or other cells can grow in together with the same environment, the media, they can all like it. So the same things for this microglia, you have another player. So we need to develop a more complicated recipe that we can make sure everybody are happy in this culture system. So that's one direction. And another direction, we are also working on this chimera model that basically we already successfully put this whole assembloid into adult mouse brain, which is of course the immunodeficient mouse. And with this system, so a lot of amazing things we already observed. First, we observe this the vessel connection between the human versus mouse, the endogenous with the transplanted one. So basically, in this model, we can for sure to recapitulate or introduce this blood flow in the human capillary, the assembly. Well, we are keep observing whether we can see other interaction, including the immune system in this mouse. So if we can, I think this is one approach that you can have a more integrated system in the endogenous in the mouse brain, and maybe also can helpful very useful for us to evaluate this functional outcome, like a whole behavioral outcome in the future, right? So, and about the last direction, we also collaborate with other engineering expert. We are trying to use the engineering approach, introduce some blood flow. If we can do that in our organoid system, we can also introduce other components, cell components like immune cells in the blood or in the organoid system. So, but all of them actually in the early stage, they are all very challenging in terms of a technique. So we are trying very hard. Yeah.
Yeah, I mean that
last point is the collaborative nature of that project is perhaps one of my favorite things about this paper. You're incorporating so many different cool approaches, different techniques from different expertise, and I think you're at a great place to leverage that collaborative nature. I mean, you're at Cincinnati Children's, and one of the people who was on that paper, the cell stem cell paper, was Ming-Sha Gu, who is of course an expert in all things vascular disease, vascular development. She's actually a mentor of mine, formerly from Stanford and Joe Wu's lab. I mean, we talk about Cincinnati Children's a lot here on the show. You have so many cool organoid approaches that you've developed, and a lot of papers that we've covered on the show, coming from Custom, the Center for Stem Cell and Organoid Medicine. Some incredible scientists there leading the charge in the organoid field, like yourself, Takanori Takebe, Jim Wells, Ming-Sha Gu, who's on the show recently. I mean, it's an amazing powerhouse in organoid biology. I mean, pick your tissue type, whether it's iPS derived or not. I'm sure there's somebody at Cincinnati Children's and Custom who are working and pioneering a new approach to study that disease. So tell us about Custom, tell us about Cincinnati Children's, what you love about working there, and maybe why it's the best place in the world for organoid biology.
Yeah, thank you so much for, again, reintroduce our customer in the Cincinnati Children's Hospital. Of course, I believe, the customer is the main reason why I decided to join the Cincinnati Children's Hospital, because this is, I believe, of course, the one of the largest, the stem cell organoid medicine center, at least in the Midwest. So with our center, basically, as you just mentioned, we have a lot of expertise in different organ genesis, including Jim Wells, including Aaron Zong, Takeba, and also Ming-Sha Gu, who is a co-author in our paper. So it's impossible if we don't have such a good center that we can develop a BBB assembloid, right? So Ming-Sha really helped us to develop this blood vessel organoid. Then we figure out how to assemble them together to mimic the neurovascular development. So I think our customer is not only we have such good resource for the collaboration. Of course, I believe we have a lot of synergetic efforts, not only for our BBB assembly, but for maybe other multi-organ system development in the future. And we also have a very unique, the collaboration with the physician in the children's hospital side. So as many of my, co-author in this paper, in other project, how we can obtain this unique, remove the tissue from the CCM patients, because we have a strong collaboration with the physician. We talk about each other, and they have a, sometimes you even don't think about is, for example, some embryos, you really unique that resource. For our basic scientists, we feel super excited that we can get access to that. We can uncover some really unknown science questions based on those resources. So yeah, the Cincinnati Children's Hospital, no matter good for the basic science, but also for a lot of these translational collaborative projects.
Yeah, I think a great template for how science is moving in this really collaborative environment, and not for nothing, but the trainees there are brilliant. Not for no reason, they invited me, of all people, to give a talk as a representative of the Stem Cell podcast, and it was one of the most delightful experiences in my life. I mean, these trainees, I don't want to call them kids, because the level of maturity was exceptional, and of course their judgment in podcasts exceptional as well. But more than anything, what spoke to me was how they did that as a culture. They had a different mindset about science than what I was used to in my training period, which was really multidisciplinary, which is easy to say, everything's multidisciplinary. But they just, in all of their thinking, they had a very firm grasp on the engineering challenges, the molecular biology, the cell biology. They had it all in their toolkit. So I think that as a place to train for all those trainees out there, to be a person learning science and incorporating that culture of science, where you need to have a lot of skill sets, you got to have a lot of people, a big team, and big dreams. So yeah, the custom crew there, they're really delivering on another level. But I can't wait for the generation of scientists that were trained there, because these are the people that are kind of, dictating the direction of everything that we're doing. So you're at a great place. You're doing great work, and you're part of that training committee. So props to you and your ambition, and it trickling down to the future of science. Before we let you go, this has been really a delightful talk. Thank you so much for sharing and your future directions more than anything. I love this show because you speak to scientists, and when they have the courage to talk about what they're trying and how tough it is, I appreciate it all the more. So thanks for sharing that. Before we let you go, though, we have a couple kind of science peripheral questions, a couple fill in the blanks that should be a snap for you. First off, when I am not conducting research, I am.
Well, yeah, when I not conduct any research, I'm really enjoy, to read the history and art books. So simply because I love these arts and, all of these beauties. So I really enjoy, to uncover some treasures, the hidden treasures. So through, visit a lot of the local antiques and, browse the website, and even visit the museum, talk to my collector friends in locally and international. So sometimes, just like as a scientist, right? So it's super excited to uncover some hidden treasures. Nobody haven't find it yet. So sometimes if you can, from many fake the fake copy, high quality fake copies, and you identify some real antiques from that ours. Wow, that's make me like super excited. So that's why I really enjoyful to looking forward to this and finding hidden treasures as I'm not working any science. Yeah. So if I'm not a scientist, I may probably to be a staff at the museum or study the history for a long time.
Yeah, Arun mentions all the time the intersection of art and science, and how a lot of scientists have that artist thread running through them. I mean, as I look at Arun behind him from his podcast studio, I see a multi-channel picture of a cardiac myocyte on the one side, and the pillars of creation. I think that's the pillars of creation on the other. So I mean, this is I think a common thread is that you see the art and beauty in your own work, and sometimes you're the first to see it, which is so exhilarating. But I think across the board, we all appreciate the beauty of not just art and science, but just the beauty in life and in creation and the imagination of all the artists out there. So I second that. Although you can't find me in a museum in the city with all the tourism. I mean, it's just out of control. It's like a zoo. Lastly, if you could have one superpower, it would be.
Well, of course, will be time traveling. I know this is like everybody, the dream superpower maybe, but if I can time travel, just like the history, the arts, I may be able to visit, some the pictures, in the history, or uncover some mystery events that we don't know, we don't have any question. But if we have the time traveling, even if I can imagine to travel back to the billion years ago, what happened to our human being development and right? So of course I'm not interpret anything when I do the time traveling. Yeah.
Well, looking back or looking forward, we very much appreciate your imagination. And I mean, one would argue that you're not just peeking into the future, but you're one of the many architects of our future success in stem cell science. So we appreciate you, the time traveler, Dr. Guo, and we look forward to seeing what you've got to share with us in the future. Until the next time, which we hope is soon, thank you again so much for sharing with us and our listeners.
Thank you. Thank you.
All right, guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including episode summary and links to all the interview and roundup papers. You can also reach out to us on X at Stem Cell Podcast or via email at info@stemcellpodcast.com with feedback or to suggest guests. A delightful conversation today with Dr. Ziyuan Guo. about all things and a roundup to boot. Wow, what an episode. We got another one coming in hot for you in a couple of weeks, though, so stay tuned. Until then, thank you so much for listening.